Welcome everyone to the last session of this symposium. This is probably one of the most exciting parts, at least for me. So my name is Nicholas Lyons. I'm the lead dairy with New South Wales DPI, and I'll be chairing this session today, the one of the Emerging Scientist competition. First of all, I want to thank and acknowledge the sponsors of this session, Hunter Local Services, for their support towards the whole broader symposium, but particularly this session today. As most of you that are familiar with the symposium and have attended previous years, either face-to-face or online, this is a key part of the symposium. It's where we get to showcase a selected group of young and emerging scientists to share the most recent research and innovation of applied insights into the dairy industry. So before we dive straight into it, a couple of housekeeping things. As usual, questions and answers need to be submitted kind of on your platform. So we'll have 7 presenters. After each one of them, we'll have a couple of minutes of questions, and then we'll bring them towards at the end together. There's a couple of minutes to do some further questions too. The other thing I want to tell you is that Niall Blair mentioned yesterday Dairy Up and the program as a whole. We are currently advertising within that program 9 PhD opportunities. So probably it's kind of a record that the New South Wales dairy industry and the Australian dairy industry more broadly has access to a suite of PhD opportunities funded by the project in a wide variety of topics. So if anybody is interested in considering a PhD, please jump on the Dairy Up website, dairyup.com.au, and there's a PhD opportunities section, and you'll be able to explore different topics and contact the potential supervisors if you're interested. Then I just want to explain briefly how this process works. So we put out a call for emerging scientists around Australia of different universities, and anybody can submit an abstract. So those that are interested in participating submit abstract, and based on that, we select those kind of tick the boxes of what we're after and show a kind of a wide range of projects to showcase in the industry. We then select the best abstract to win a prize, and we'll announce that towards the end. The group of selected people that will participate then need to submit a 1 minute promo video. So that video was available on the Facebook page. Some of you might have seen it. Great work of the 7 emerging scientists, getting more and more creative as we go. We also select the best promo video, and again, we'll announce at the end. And the last bit that we're kind of going to be judging is the actual presentation that they will be delivering today. So each emerging scientist will have 5 minutes to present the research. We'll tackle questions. After the 7 emerging scientists present, at the end, you as an audience will be able to select who's the emerging scientist that you think deserves winning this oral presentation of the Emerging Scientist competition. So that's really exciting, and you get to be part of it. So without further ado, I want to thank also the scientific committee for all the effort on judging and selecting the speakers today. I'll dig straight into the first presenter. We've got kind of an international experience either because they were born somewhere else or they've explored overseas to do some of the research or the work in the past within our 7 speakers. So our first speaker is Santos Amadeo. Santos is originally from Argentina. He did some work in his grandparents' farm in Argentina, worked for some farms in South America too. Always had a passion for agriculture, and probably his interest, a particular kind of attraction, was around the deterioration of the natural resources around agriculture, and how can we explore kind of leaving a better environment for the future generations. And that brought him to Australia to deepen his studies in sustainable agriculture. So Santos will be talking today on giving dairy farmers extra motives to reduce greenhouse gas emissions. So without further ado, Santos.
Thank you for the introduction, Nico. Very well. Okay, good morning, everyone. Today I'll be talking about optimizing the carbon trade. That was the name of my paper, or as I chose to name it today, giving dairy farmers extra motives to reduce greenhouse gas emissions. Briefly, the paper was set in a dairy company with 2 dairy farms. I chose this as it was my current employment when I chose the topic. And the objective of the paper was to find out whether it was possible to lower greenhouse gas emissions and the cost of production by optimizing the processes between the 2 farms. Once I could prove this was possible, I wanted to see whether it was possible to extrapolate it to larger scale in regions or catchments. I used the Australian dairy carbon calculator to calculate greenhouse gas emissions from both farms and the whole company. So one of the focus points on my literature review was the role of agri-environmental schemes as facilitators. How these schemes help farmers transition to conservation agriculture and biodiversity conservation. Of this list, I focused mostly on the last scheme, which is the Emissions Reduction Fund, also prior known as the Carbon Farming Initiative, in which I unearthed some of the problems, barriers of adoption, and the possible solutions to get more people to participate in it. Given the dairy company I work for is a small farm, I focused also on what were the barriers of adoption for small farm holders into the Emissions Reduction Fund. Here we can see an example of an aggregation under the Emissions Reduction Fund in which different farm units add each other's carbon credits into an aggregator in order to increase the returns of carbon credits and decrease the cost of the project of participating in the Emissions Reduction Fund. Studying into it, I realized it's very much a simple addition of these sites, very much a linear model, and no integration between the farms. So given the setting with the 2 dairy farms of the company I work for, I thought, well, wouldn't it be better if these aggregations would instead just being additions could integrate and cycle the carbon sources that contribute to the greenhouse gas emissions in order not only to lower greenhouse gas emissions, but to lower the cost of production and also get a higher return in Australian carbon credit units. So if this was possible at a small scale, why could it not be possible at a larger scale? One of the examples or one of the methods that dairy farmers can participate in the Emissions Reduction Fund is the animal effluent management. This is just a graph showing how an anaerobic digester works. In this case, livestock waste from effluent and wastewater could be converted into biogas or biomethane for energy. And the result of that could also give farmers the opportunity to tap into natural fertilizer and soil amendments of organic nature, of course, in which they would be using their own nutrient cycling in order to, well, grow better feed for the animals and reduce the cost of inputs of urea, common nitrogen fertilizer. So here in this slide, I just compare what a linear model dairy farm looks against the circular dairy farm model. On the left side, we see how inputs are high and the outputs are many. While where we see the right side, the inputs are lower and the outputs is just the milk. We reutilize on this side the greenhouse gas emissions, the waste, and the nutrients all get back and cycled in order to reduce greenhouse gas emissions and the cost of production, as previously mentioned. So my project was a dissertation submitted for the Masters of Sustainable Agriculture coursework. It was small in scope and time availability, so the Australian Dairy Carbon Calculator didn't end up allowing me to fully test my hypothesis. So this table just shows the result of different scenarios in which I trialed to see how these two dairy farms and the whole company could apply into the emissions reduction fund opportunities. To wrap up, although my conclusions didn't prove entirely my hypothesis, I still believe it is very much doable. And I actually learned recently it is being done somewhere here in Australia, which I'll look into right now. Before that, I just wanted to share this quotation from one of the papers I got into the literature review. Let me see if I can move this. Understanding how innovation, learning, and knowledge sharing are interrelated and can induce sustainable practice change in a farmer-driven environment is essential for advancement of knowledge and how to collectively transform our relationship with the earth, particularly when it comes to farmers who are often depicted as individualistic and neoliberal. This was my presentation, and that is all. I'm open to questions and answers. Oh, here is the hypothesis and practice. These are a couple of links which I'll share in the comments so you can tap into. It's a project being done in Narra in the south coast of New South Wales, where 19 dairy farms are actually doing my hypothesis, in which they're connecting through pipes all the effluent into a central facility for bioenergy production. Sorry.
Perfect. Thank you, Santos, very much for that presentation. And definitely, I think it's a very topical issue. And oh, sorry, you had another slide?
No, sorry. Finish right there.
No, that's fine. I think it's a very topical kind of topic, and something that I know industry is looking forward. So I've got a couple of questions that have come through. One is, can farmers use the calculator to plan how they can work towards a more carbon neutral system?
Yes, of course. One of the perks of this Australian Dairy Carbon Calculator is that it gives you abatement strategies based on your emissions. So it's very complex Excel sheets in which firstly you calculate the baseline farm greenhouse gas emissions, and according to what the emissions are, you following sheets would show you what the better abatement strategies for your case are. So yeah, it's quite a useful tool.
Perfect. Then there's another question that came through. Remember, you can type your questions in the Q&A box. There's one that says, in your modeling, did you have a look at the contribution that replacement rate makes to emissions?
Yes, to some extent, because of the nature of the Australian Dairy Carbon Calculator. It's a protected sheet of Excel, so I couldn't play too much with the different scenarios. So when I tried to calculate both farms together as a whole, for example, it didn't allow for proper calculations to be done and subsequent abatement strategies to be developed. But it is somewhere inside the large dissertation.
Perfect. And probably the last one at this stage, and we might come back to it towards the end, is, do you have any insight of comment around the kind of the willingness or the barriers for farmers to engage in these kind of things, either individually or kind of aggregated?
Yes. Well, as I mentioned in the literature review, I went deeply into what the reasons or barriers of adoption to the different agri-environmental schemes are. And there are many, of which the most interesting to me and many researchers came in accordance with is the culture of barriers. Many farmers thought that the cultural side of applying into conservation methods might be not well seen from neighbors. Other reasons might be the loss of yield reduction of yield and profitability. Some others are, especially in carbon markets, the volatility and the still untapped nature and newness of the carbon markets gave farmers a bit of hesitance when applying to them. So yeah, there are quite a few barriers and reasons why farmers are not fully invested into the agri-environmental schemes.
Perfect. I think work like this helps untangle a bit of those messages and get a better insight. And it's definitely, as I said, a topical issue. So as an industry, we need to continue to look for those opportunities. So, well, thank you very much, Santos, for your research and your presentation today. We'll hold on till the end to see how you go. But I want to thank you again for the abstract, the video, and your presentation today.
Thank you.
No worries at all. So with that, we move to our second speaker today. Our second speaker is Sita Ritchie. Sita has a vast experience working in the dairy industry, both in Australia and overseas, with stints around Indonesia, Pakistan, Philippines, Ethiopia, and the likes. Sita is currently a development officer dairy within the New South Wales DPI. And she has a vast experience in farm business management, climate risk management, and this research was part of a collaborative project within DPI. Sita's talk is titled Environmental Consequences of Replacing Dairy Production in New South Wales. Sita, the stage is yours.
Thanks a lot, Nico. Thanks, everyone. So today I'll be talking about the environmental consequences of replacing dairy in New South Wales. And this is some emerging research that we've been working on in DPI, some preliminary results that I'd like to share. I'll just go along next. Why is it not working? There we go. So just to set a bit of context, I guess. Look, we're probably all aware, and it was great to have Santos's presentation first, because we're all aware that there's global efforts to reduce greenhouse gas emissions globally. And the Australian dairy industry has set targets really along these lines, especially under the Dairy Industry Sustainability Framework, to reduce greenhouse gas emission intensity by 30% by 2030. So as an industry, we're working on this issue to try and reduce emissions. And typically carbon and water footprints have been used to kind of quantify the environmental impacts of the dairy industry. And look, these footprints have often resulted in calls for consumers to reduce dairy consumption to minimise their impact. However, what footprints don't really account for is the consequences of changing or reducing a certain type of production, and the consequences of the impacts in supply and demand of filling that demand if you cease one production and shifting the impacts to somewhere else. And this is where this analysis really comes in. So what are the consequences if we cease dairy production in New South Wales? We were looking to see if we stopped all dairy production in New South Wales, what would be the actual impacts of this, given that there's probably something that will come in its place. So we looked at replacing dairy in New South Wales with an equivalent, so replacing dairy products and co-products. So in this case, we looked at plant-based equivalent, which in this case, soy. So we looked at replacing milk, cream, red meat, and other products with soy-based equivalents. And we chose soy because we wanted to use a crop that would be most suited in areas of New South Wales that wet dairy are currently being produced, so in the north and south and inland. We're trying to produce as much of that in New South Wales without having to go outside of the state. So that's why we chose soy for this study. So what is the carbon footprint of dairy? On an annual basis, we looked at 1 year and we estimated that the annual emissions to produce dairy in New South Wales is 1.15 megatons of carbon dioxide equivalent. So this was a cradle to gate type analysis where it's looking at all the emissions in New South Wales embedded in producing that milk. So this also includes the inputs that we use on farm, right up until it leaves the farm gate on the back of the tanker. So then we looked at, okay, so if we cease dairy production in New South Wales and replace it with soy, what would be the impact? And what we found is that we're likely to see a reduction in total emissions if we transitioned away from dairy completely and moved to soy of 0.75 megatons of CO2e, which is a lot less than what a carbon footprint suggests. On the previous slide, it was 1.15 megatons. From a footprinting perspective, this would indicate that if we cease dairy, we'd get a reduction in 1.15. But actually, if we're looking at thinking around the respects that would replace it with something else like soy, we only get a reduction of 0.75. But what's more interesting is that to fill that gap in dairy, we'd actually need additional land outside of New South Wales, up to like approximately 66,000 hectares, extra additional agricultural land needed to grow soybeans to fill that demand with an alternative product. So that's quite significant, and it's shifting that agricultural burden, you know, somewhere else, which is not often thought about when we're just looking at the footprint alone. We also found that there's no significant water savings as well. So although if you cease dairy and reduce water, that water would be then diverted to other agricultural activities. So the water savings there aren't as significant. So then we thought, well, what are the mitigation options? And Santos also touched on some of the options to reduce emissions on a dairy farm. It's like, well, as an alternative, maybe it's better looking at dairy and looking at a couple of mitigation options. And then what would happen if we implemented these strategies to reduce emissions and cease dairy production compared with soy? So we looked at two options, capturing methane from effluent ponds and also looking at alternatives, so additives, so Asparagopsis, which is a red seaweed, which has proven emerging research to show that it reduces methane emissions. So if we applied these two strategies across the New South Wales sector and then compared it with soy, what would be the difference? And interestingly, that if there's potential there, if dairy implemented these two strategies alone and we compared to soy production, it would actually increase total emissions if we shifted from dairy with mitigation to soy production by 0.31 megatons of CO2e, which is really quite an interesting result. So I guess in summary, the key messages that I wanted to leave today was that looking at environmental footprints alone don't account for changes in demand, and altering consumer demand, you know, on the basis of footprints alone will not result in expected, you know, carbon savings often. So although these results show that ceasing dairy and replacing it with soy in New South Wales could reduce emissions, it also shows that additional agricultural land would probably be required globally outside of New South Wales to grow those additional soybeans to replace what's been lost, I guess, from dairy. And it's unlikely to increase water availability to the environment. So I guess this just leaves us with an opportunity of exploring what the potential for dairy around mitigation options, rather than looking at a replacement and a replacement in that sense. And I guess as a comparison, looking at dairy and the opportunity for soy, and that really just removing or ceasing or reducing a certain industry, there's impacts down the chain by doing so. So I think that's the summary of the research. And if you'd like any more information, feel free to email myself or Aaron, who's been the lead scientist on this project. And yeah, happy to see if I can answer any questions. Okay, thanks, Nico.
Thank you, Sita, for that presentation. And it's really exciting, as I told Santos before, that an issue that is really topical and that as an industry we need to address, and we are addressing, that there's 2 out of 7 emerging scientists today talking about it. So it's really exciting to see that. I'll encourage you to send questions if you want. There's questions that you can flick through. The questions that came through were around, you mentioned effluent and additives. Any comment of why did you pick that, and what other options could be out there?
I guess my understanding is why Aaron chose those 2 parameters was, I think, the biggest bang for your buck. Where could we save the biggest amount of the claims? I guess a lot of the emerging research with additives is it can reduce, you know, methane by about 80% with that particular additive. So we thought we'll just try those two strategies to start with, but in saying that, there's a lot of other opportunities to reduce emissions out there that weren't included in this study. So nitrification inhibitors, energy, renewable energies on farm, all of the suite of other options out there, which wasn't included in this study. So you could speculate that if all of those other mitigation strategies were also implemented, there could be even greater scope to reduce emissions. And where you weigh it up against soy would be have a way less environmental impact than soy in that particular instance. So I just for this preliminary study, we just focus on 2 additives, and then for sure, maybe down the track, we can explore others as well.
Perfect. There's a comment, good work, Sita, and looking at the full picture is always important. Good to see more of the cost benefit of those mitigating strategies. Maybe a comment of maybe our responsibility as an industry of telling this full picture. Like, do you have any thought of kind of, it's not only about the footprint, it's about the whole consequential analysis of doing one or the other? Yeah, any reflection there?
Yeah, I think it is, because I mean it's easy for, as a, you know, I think as a consumer, or I guess trying to think at things quite linear, you know, in a linear way. It's like, well, if we stop doing this, therefore we're reducing an impact. But it's not actually that straightforward, because that demand is still there. So that demand has to come from elsewhere. So if we're stopping an activity in one area, it's having flow-on effects down the chain. So I think as an industry, I think that's an important message to sell. I think as our responsibility as an industry to promote that positive image, and and promote the things that we are doing as an industry in dairy around environmental sustainability, which is good for the industry in the future. Yeah.
Perfect. There's another question that came through saying, Sita, nice piece of work. Have you thought to also add nutrition to your analysis? Because they're very different value in the nutritional quality of these two products.
Yeah, in terms of from a consumer perspective, yeah, very
probably,
interesting point. Not at this stage, but I take that as a note. I mean, there's so many variables that you could look at this. I mean, other questions have been raised. It's like, why didn't you look at almonds and or oats and things like that? I guess for this initial preliminary study, we thought we'll just focus on one other key commodity that we think could be more easily replaced with dairy. But there's so much scope there, particularly from the nutritional side. That's a very good point. Yeah, which hasn't been considered in the scope of this work yet.
Yeah, but it's good to see. Obviously, we've got an intrinsic interest in the dairy industry by obvious reasons. But it's good to see that it's about telling the full story of the footprint, and that eliminating is one way. But in fact, if we can address the issues by mitigating, we can really be in a much better position. And that it's not only around milk. The last question is probably: have you considered offsetting with trees? And any option, any comment there?
Yeah, actually, that's interesting. So a bit of that was actually already incorporated in the analysis. So when we're ceasing dairy, Aaron looked at, well, where's the milking platform versus support area, and some areas aren't suitable to grow soybeans, in which case those areas were planted with trees, and did modelling of that way. So trees were actually taken into account of the modelling in this analysis too. But again, I guess as in terms of offset for dairy mitigation strategies, that's another way to look at it as well, which wasn't included in the study. Yeah.
Perfect. Sita, thank you very much for your time and your insights, and again for the abstract, the video, and your presentation today. And we'll hold on till the end. Remember, it's you, the audience, who's selecting who's the best presenter today. So please keep your tally and how you're scoring them, and we'll vote towards the end. So we'll go now to our next speaker. Juan Ignacio Gargiulo is originally from Argentina too, so we're seeing here a trend going on. He did his Bachelor of Ag Science and a postgraduate degree back in Argentina, and has just completed his PhD at the University of Sydney, working within the Milking Edge project around applications and impacts of automation in pasture-based dairy systems, and he's currently also a development officer dairy within the New South Wales Department of Primary Industries. So Juan, the stage is yours.
Thanks, Nico, for the introduction. So I would like to start by defining what is a decision support tool. And basically, it's a computer-based system that helps farmers use data, models, documents, or knowledge to solve problems and make decisions. So why the industry needed a tool for automatic milking systems or robotic milking systems? And the first reason is that these are really complex systems with many variables interacting with each other. And we identified that there were no tools to assess the impact of changes in economic and physical variables for automatic milking systems. And we thought that such a tool could be really beneficial for farmers considering investing in the technology, because it could help them to better plan the system and reduce the uncertainty in the investment, and also could be really beneficial for farmers currently operating with the technology to help them to identify areas of underperformance and also optimize the system performance, actually. So actually, we worked in the Milking Edge project in the development of the first decision support tool available for pasture-based automatic milking systems. So this tool is based on 5 years of physical and economic data that I collected as part of my PhD, mainly based on 28 international AMS farms from Australia, Ireland, New Zealand, Chile, and Argentina. And also it is based on 18 more Australian farms, more economic data collected with the dairy-based methodology and dairy farm monitor methodology. So this tool, we build it in a user-friendly interface, in a web-based interface. So the tool allows the user to simulate scenarios. For example, here the user can change the cows in milk and see the impact of those changes in other variables. The user can also set target for key variables and run optimization simulation to see how to go from the situation A to the situation B. And also the tool allows the user or the farmer to benchmark the performance, the current performance or the simulated performance, and compare it with commercial farm data. That is the data that we collected in all those years. Another key feature of this tool is that we tested or evaluated the accuracy of the predictions, and we used 21 AMS farms and 2 years of data, also from Australia and overseas. And we found that overall the accuracy of the predictions was really good. There were a difference between the actual and predicted values between 5 and 10%. Sorry. Also, we tested the perception, the farmer's perception of this tool. Basically, we tested how useful was this tool and how easy to use was this tool. So we tested this with 11 farmers, also from Australia and overseas. We allowed them 1 month to test it, and we then sent a questionnaire. And overall, they rated the tool in usefulness and ease of use 4 out of 5 stars. So the tool was really well received by the industry, and we launched it in August this year. And as you can see here is the users that used this tool since August. And we had so far more than 350 users from across 30 different countries that use this tool, and it's still growing. It also caught the attention of the media, and different articles were published in different magazines on the tool and how to use it and what's for. So in summary, this research is the world's first decision support tool available for pasture-based automatic milking systems. The main thing of this tool is that it's based on unique farm data that we collected across multiple farms and years. There were few tools available and were mostly based in models and assumptions. So this is really unique because it's based on real farm data. And you can access this tool in the link below. I can share it later in the comments. And we really will appreciate your feedback and comments on the tool, that you can leave it there in the website. So thanks for your attention. I'm really happy to answer any questions.
Perfect. Thank you, Juan, very much for that presentation. Remember, to all the audience, just a reminder that you can type questions on the Q&A tab as we go. There's one question that came through saying around, it's very interesting that you've got a kind of uptake or interest from different countries. Is there an issue that it's for different type of systems or size? Or what type of systems does the tool cater for?
Well, as I mentioned, we mostly use pasture-based systems. Actually, it was developed with pasture-based systems only, but as I showed, the attention of the usage was also in North America and Europe. So, but the concepts and how the variables interact could be useful also for other systems. But it was mostly designed using pasture-based data, and yeah, with a range of size that represents well the Australian dairy industry in general, around 200 cows on average for AMS.
Perfect. There's another question that came through around Does the tool allow for prediction, or just it provides information about the data?
No. So we use the tool mainly to develop some models that are the predictive models. So you can predict things with the tool. You can predict economic and physical scenarios, but also those colors in the gauges that I show allows you to compare with the data that sits behind the tool. So for example, if it is red, it shows you that that performance that you are simulating or predicting is in the lowest or the bottom 25% of the farms. So you can, it works with models that are based on, that we develop based on the data collected, and also allows to benchmark against that data.
Perfect. So there's kind of a follow-up question of how into the future does it allow you to predict some of that data?
So can you repeat it?
Yeah, it says you said the tool allows you to predict what are some expected values.
Yeah.
Is there a timeline in that prediction? So it predicts out to a certain amount of time, or it predicts kind of one point in time?
No, it predicts one point in time at this stage. Probably it could be expanded to predict across time, but at this stage it predicts only one point of time. So you input some variables, some physical variables, for example, and predicts, for example, the milking frequency, or predicts expected return on total assets or EBIT based on the variables that you enter. But it's just one point in time.
Perfect. And then there's another question that says, do you have any feedback of the people that have used the tool out of those 350 people on the use of the tool itself? Could this tool be expanded to other technologies or other systems?
Yeah, so not formally, of course, but we got informal feedback, and it was really good from different service providers and different farmers that tested. And well, as I mentioned, we formally tested with a questionnaire and a model or a framework that is designed to evaluate the usefulness and ease of use. And well, it was mainly what we got in the feedback when we launched it, but it was similar, that it was easy to use and useful for farmers and service providers, also consultants that want to have a discussion with the farmer also.
Perfect. Well, Juan, thank you very much for your presentation today. And again, once again, for the abstract, the promo video and your presentation. And we'll hold off till the end to see the final results.
Thanks, Nico.
No worries. So now we jump on to our next speaker. Surprising, this person is not Argentinian, given the trend that we are seeing at the moment. So Lara Howen is a 4th year's honors student studying animal and veterinary bioscience at the University of Sydney. Although she grew up in the city, she's had a great passion and interest for animals, and has seen herself attracted to the dairy industry and a career around the dairy industry, particularly around calves. And she's currently doing her honors in dairy science in this region in a large case study. And Lara will be presenting on monitoring calf health using an accelerometer-based ear tag. Lara, all yours.
Thanks, Nico. Perfect. Can you hear me okay? Yeah, great,
Yes.
Fantastic. Okay, so we'll make a start. So as you know, calves are very important as they go on to become the future producing dairy cows of the industry, but we've got a problem on our hands. So calf morbidity and mortality is currently high, both in Australia and worldwide. So the rates in Australia were actually above the industry targets set by Dairy Australia. We're meant to have less than 10% morbidity and less than 3% mortality. So of course, calf morbidity causes major economic losses to farmers. So we've got that decrease in production because sick calves don't grow, and then that will affect their age at first calving and their milk production rate in future. We've also got vet treatment costs as well as calf loss. So we do need a solution. So technology has the potential to detect disease potentially more accurately than human assessment. It can provide continuous monitoring, early disease detection, as well as health alerts. So our study is using the Alflex ear tags that monitor activity, rumination, and suckling, and that'll generate a health index to estimate the health of calves. So it sounds promising, but the system hasn't yet been evaluated in young stock. So that's why the aim of my study was to evaluate this ear tag to detect calf-reduced health, and we're comparing that to visual health checks. So for my study, we enrolled 50 calves over a 2-month period and attached the Alflex eSense ear tag. We took daily visual health checks on all calves during morning feeding, and I'll explain a little bit further about that on the following slides. But we also recorded their feed consumption and observed if they drank all of their milk. And we also took rectal temperatures twice a week, as well as if they were visually observed to be sick. So these were the health parameters that we recorded. They were scored a 0 if they did not have any presence of these clinical signs, and a 1 if there was presence. We had a low disease prevalence on the farm overall. The most commonly observed clinical signs were scours and high temperature. And then for the technological monitoring, that relies on the suckling, rumination, and activity data. It puts it all into an algorithm and generates a health index. So the calves will start at 100 in good health, and then this number depletes as the calf becomes sick. So it gives out 2 measures of calf health. We've got below 85, which we're deeming health events. That's more of a warning. And below 76, which is health alerts. And that tells the farmer to act as soon as possible. So looking into what specific clinical signs might be the best predictor of a health event or alert, we found that the scours and temperature were the only clinical signs that had a true effect. And that wasn't really surprising considering they were the most commonly observed clinical sign. But we found that calves with scours were 3.8 times more likely to receive a health event than those without. And similarly, calves with a high temperature were 7.4 times more likely to receive a health event. For the health alerts, it was only temperature as the clinical sign that had a true effect, and they had 3.7 times more likely to receive a health alert than those without. So it's evident that these ear tags have an association between rectal temperature and the occurrence of health events and alerts, despite the tag not actually having a specific ability to measure body temperature at this current moment. So we found that this was a major benefit of the tags, as we know rectal temperature has long been considered a gold standard of disease detection, but it is incredibly time consuming and labor intensive to be taking rectal temperature every day. And as a result, most farmers never will. So if these tags can predict that calves are sick with a temperature, then it means that there is potential for it to be a good tool to aid in that disease detection. So another cool finding we found was that calves that were receiving health alerts also had a lower feed consumption than those without alerts. So specifically, for every kilo increase in feed consumed, the odds of a calf receiving a health alert dropped by 80%. So that might be an indication that the calf is not feeling too well if it's not eating as much food as it usually would be. So what is it that we can take away from all of this? Well, we saw that temperature had that strong association with the health events and alerts. And we also identified that the sick calves are eating less feed, and that this was also reflected in the health alerts that we were getting. But these tags were picking these things up, but it was only a preliminary study. So we really should be conducting some further research, particularly surrounding a validation study, so we can take hourly measurements and match that up with the tag outputs, because we only did daily measurements. And we'd also want to do this on a farm that had a high disease prevalence, as I mentioned, we only got low measures for the majority of the clinical signs. So doing this would provide us with a better understanding of tag performance, and therefore give us the ability to make better recommendations and more robust conclusions around this. And I just want to acknowledge that this study was funded by Dairy Up Program P6, and also big thank you to Alflex for supporting us and supplying ear tags, and Amanda Doty for working with us on this project. And I will take any questions you might have now.
Perfect. Thank you very much, Lara, for that presentation. Really interesting studies. Remember, you can flick questions as we go. The first question, Lara, is around, thank you very much for your presentation. How many calves were in the study, and what was their age range?
Right, okay, so we had 50 calves on the study, and their ages were right from birth up until about 60-ish days of age. So this is pre-weaned dairy calves.
Perfect. There's another question saying respiratory diseases are quite common in dairy calves. Why didn't you find any kind of positive results in there? Is it due to the low numbers that you mentioned at the end?
That's correct, yes. So basically most of the clinical signs that I included in the scoring sheet were to include those that relate to respiratory disease and any sort of enteric diseases as well. But we did find, yes, that's exactly the reason, is because we didn't have very high numbers. So we had at that farm a higher outbreak of the enteric diseases, the scours, and not so much with the respiratory diseases. So we couldn't find any clear links there.
Perfect. I've got a question myself. Was there any, can you make any comment on the timing of those alerts compared to the clinical signs of those events, particularly scours, obviously?
Yeah, that's right. So we wanted to look into this further. Obviously, just had a little bit of time limitation because it is an honest study, but we did want to look at a lag analysis. So looking at whether those tags are coming the same day that the calf is receiving those clinical signs, or is it the next day, or is it even before? Like, do we have any ability to predict some of those clinical signs before they occur? So we didn't look specifically at day before or day after, but for the temperature in specifics, that one was very strongly correlated with when they had a temperature on that day. Yeah.
Perfect. Then there's a comment in the chat for the source of those graphs that you showed, mortality and morbidity. So probably if you can then share in the chat the source for that would be good.
Yeah.
There's another comment saying, were the tags used standard tags and algorithms used on the milking herd? Do you think Allflex would consider developing new algorithms specific to calves?
So this one is specific to calves, specifically individually housed calves. So it's through an application called Young Stock, and that one is a specific algorithm for those young calves, in those individual housings. Yeah.
Perfect. And then there's a last question that says, did you observe any clinical signs before the temperature rose in general?
No, so it was most often to be temperature first before those clinical signs. But obviously I wouldn't want to make any further comments on that because we didn't have any high disease prevalence. So it was mostly just the temperatures and the scours that we were getting, and they were either coming hand in hand, or we were getting temperatures first and then observing the scours afterwards. Yeah.
Perfect. Lara, thank you very much for your time today. Again, as I tell everybody, thank you for the abstract, the video, and your presentation. And we hold off till the end to see the scoring from the audience.
Perfect. Thank you.
No worries. So we jump now to our next presenter, Beth Scott. To those that have been attending the DRF Symposium before, Beth will be a well-known person. Beth was an emerging scientist when she did her honors studying at the University of Sydney, but she's come a long way since then. Growing up on a dairy farm, studied at a Bachelor of Animal and Vet Bioscience at Sydney University, went on to do her Masters of Animal Science in Animal Breeding and Genetics in Wageningen University in the Netherlands. And she's now a PhD candidate studying inbreeding and dairy genetics at La Trobe University at the same time that she works part-time for Dairy Australia, involved in the Genomic Acceleration Project. So Beth's presentation is called, Does Selecting for A2 Milk Increase Inbreeding? Beth, all yours.
Thanks, Nico. Legacy bulls, influential bulls that continue to have an impact on the dairy industry for the following generations, even well after they've died. Probably the most recent legacy bull is Ensenada Taboo Planet. You might have heard of him or some of his sons. Only the 9th bull in the whole world to sell more than 1.5 million doses of semen. He's A2A2, and he has more than 90 sons in AI and 250 grandsons in AI. That's just in Australia. But can he be in too many pedigrees? Is his influence too big? Well, we know that intense selection, say for this bull Planet, can cause an increase in inbreeding. Inbreeding can result in the loss of genetic diversity, a decreased response to selection, and ultimately decreasing farm profitability. Irrespective of selection for a specific bull or a specific gene, the introduction of genomic selection has seen an increase in the rate of inbreeding per year in the Netherlands, France, USA, and here in Australia. But this could be worse in the case of polled, as the frequency of cows carrying 2 copies of the polled gene in Australia is less than 1% in the genotype population. But it could also be a risk when the allele is more abundant, as with A2, due to its increased selection since the development of the A2 cooperation. Unlike polled, however, herds breeding for A2 have to be entirely A2. But why do we actually care about reduced animal performance from inbreeding? And as a farmer, is it something that we should be worrying about? Based on Australian data, we know that a 1% increase in genomic inbreeding results in a 0.63 kilo loss of protein. So if we take a group of cows that have good genotypes and performance records, we know that under genomic selection, they've been able to increase their protein breeding value by 4.5 kilos. But their genomic inbreeding has also increased 1.15%. This results in a net gain of protein of 3.7. But we also know that inbreeding affects traits worse in traits such as fertility and survival. Now, we're seeing these reduction in performance without selecting for something specific, such as A2 and polled. So let's talk a little bit about A2, which is where I focus my study on. A2 falls on chromosome 6 in what's called the Casein Cluster. There's 4 genes here, which account for approximately 75% of the milk protein content. I looked at the frequency change of A2 over an 18-year period. And I found that the A1 cows in the national herd, in about 46,000 cows that had genotypes, that it decreased from 13 to 8%, whilst the A2 increased 20% in the same time period, which is unsurprising, but quite interesting considering less than 50 farms out of 5,000 farms have a contract for an A2 milk, which these results suggest that most farms are heavily selecting for A2. I also wanted to check if there was actually any big differences between the 2 animals. Is an A2 animal performing better than an A1 animal? And the answer was sort of like roughly no. They were slightly better on the Balanced Performance Index. But A1 animals had better performance for health traits such as fertility and somatic cell count. However, when we zoomed in on chromosome 6 and looked at the inbreeding using a novel method, we found that these animals were 2% more inbred than their A1 counterparts. And if I then looked at young animals, so animals born between 2013 to 2017, this difference stretched out to 4%. It is likely going to be a similar story for polled, but potentially worse, as less than 1% of cows are currently carrying 2 copies of the polled, whereas 32% were carrying A2 when they started selecting heavily for it. There is very few bulls that are high BPI and polled, and it's also located on the same chromosome that we know causes early abortion in Holstein cows. So in summary, my research found that A2 animals were more inbred than their A1 counterparts. Heavy selection for A2 could be reducing milk protein production. Given that few farmers are actually getting paid for a premium for A2 milk, is it really worth having a strong selection on it in your herd? If I can give you one takeaway message for farmers listening in on this, it's to run your bull team through an inbreeding program, whether it be through your bull company or your herd test center. It takes less than 5 minutes to make sure that you're using the best bulls for your cows. Thank you.
Thank you, Beth, very much for that. And again, a topical issue. Again, I encourage all the attendees to send questions if you're interested. There's one question that has come through. It says, interesting presentation. Is there a way to sustainably select for A2, A2 or polled without increasing the level of inbreeding?
I think inbreeding is going to increase regardless because the dairy population is a limited population size now. But if you make sure you are using the most diverse bull team possible while still meeting your other breeding objectives, yes, there is a way. I'm looking at developing methods that use genomics to manage the inbreeding, which is a more accurate way and can determine differences between full siblings, for example.
Perfect. There's another question saying you mentioned there was 50 farms that had a contract for A2. Is this problem going to get bigger or smaller if more farms sign up a contract for A2? And what proportion of the national herd is A2?
So 52% of the genotyped national herd is A2, which has increased the 20%. Whether it gets bigger, it depends if people start taking inbreeding seriously. If people start to manage inbreeding, you literally just have to send your current cow list to a herd test centre and they can check if your bull team will cause a significant increase in inbreeding. Perfect.
There's another question saying, what's the industry doing as a whole to address increasing levels of inbreeding?
My whole PhD is on inbreeding, and there's a whole project within my group to look at ways to manage inbreeding. It's quite a complex issue where there's multiple levels that have to work together. So the farmer can work with the herd test centre, the breed societies can work together, but it requires a full level of integration to sort of actively manage it, which is quite a complex thing to implement. But hopefully at the end of my PhD, I'll have something that can better manage inbreeding for all Australian farms.
Hopefully it does, and then you can ramp up to full-time to start sharing these insights. The last question is, you mentioned polled genes and A2A2. Is there an issue, a potential issue with other traits?
I don't quite understand that question.
I think the question taps into, you mentioned looking at polled kind of bulls and A2A2. Is there any other thing that could create an issue with inbreeding moving forward that we select for?
I think those two are the big ones. Like if you think about, if you select for A2 rather than straight protein yield, it will likely increase inbreeding at a faster rate than just protein yield. So same with polled, it's a single gene. So it increases inbreeding quite fast, especially if you're really trying to get those high genetic merit bulls. And there's quite a few, there's not many that come from different cow families or bull families that are available. So just managing those two, but I think those two are the two big flags. But if you're selecting for a specific gene, as opposed to a trait that has multiple genes, that's when the inbreeding starts to really stack up quickly.
Perfect. Beth, thank you very much for the abstract, the video, and your presentation today. And I'll see you towards the end.
Thanks, Nico.
No worries. So we'll now jump on to our next emerging scientist, Sarah Mack. Sarah Mack might be known to you too because she's presented before. She's a third-year PhD candidate at the University of Sydney with an experience that ranges for over 6 years in the dairy industry, both in Australia and overseas, as a farmhand, but also as a researcher. And she's doing projects around looking at the challenging of longevity and cow-calf systems and weaning methods in the dairy industry. So Sarah, all yours.
Thanks, Nico, for the introduction. So this study that I did goes alongside Laura's study. However, I did focus more on the development side and the impact of weaning. So I'll be presenting on monitoring calf rumination development using an accelerometer ear tag. Uh-oh. Not good. Current weaning methods in the dairy industry are based on age, weight, and feed consumption. However, this doesn't give a clear indication of what's going on in the rumen. The objective of the study was to determine the variability in rumination development between individually housed calves on a set dietary regime from birth to weaning. Now, this is a foundational study, as once we understand the variability, then we can apply this to a weaning method focusing on rumination, because rumination is a direct link to rumen function. The two measurements I will be focusing on today are the daily rumination based on the ear tag, and then the daily feed intake based on the total mixed ration. Okay, so there's a lot going on in this, so bear with me, we'll get through it. On the left, we have our feed consumption in kilograms per day. On the right, we have rumination in minutes per day. And on the x-axis, we have age in days. Our orange line is feed consumption, whereas our blue line is rumination. When we look at this green dot, this is our first rumination, and that's defined by the first day that we find the calf ruminating over 5 minutes per day. However, we can see that the average is actually about 25 minutes per day, and it's around day 8. These black lines signify our variability in age in which we first see this rumination. Our consistent rumination is defined as 3 days of 5 minutes per day or more. And we're seeing that happens around day 12 and is an average of 48 minutes per day. When we look at our peak, pre-weaning peak, we can see that there's high variability in the days of age in which we see our calves reaching their peak rumination. However, the average is about 37 days. This gray dot is rumination around weaning, and our orange dot is our post-weaning peak at 56 days. This red line signifies weaning around about 54 days. When we look a little bit more in depth and we compare our first rumination to our consistent rumination, we can see that our consistent rumination has high variability compared to our first rumination in minutes per day. However, this is expected as the first time we see the calves ruminating, they are at a younger age. When we look at our peaks, we can see our pre-weaning peak as well as our post-weaning peak are a bit more condensed when it comes to the minutes per day that the calves are ruminating as compared to weaning, which is a bit more variable. However, we do have one outlier for each time period. Now, this is the same calf, so I looked into what's going on with this calf over here. This calf actually was quite sick at the beginning, and we can see that this impacted the rumination development. However, at weaning and post-weaning, we see that this calf here actually meets the averages of our herd. This calf, however, has an opposite story. This calf was in the average of pre-weaning and then decreased significantly in rumination per day for weaning and post-weaning. Now, what we can take from this is that our rumination of our pre-weaning does not necessarily signify our rumination from weaning and moving forward. Now, going back to our graph, we have for the first 40 days, we can see that rumination increases irrespective of the feed intake, which is quite interesting. And honestly, this has brought up so many different questions that we're excited to kind of dive into our data and be able to implement that into an individually based weaning method based on rumination in order to allow each calf to reach their optimal potential for production later on in life. So thank you for listening. I would like to thank Dairy Up for sponsoring this study. And like Cameron said, this is under P6. And then also I would like to thank Amanda Doty from Allflex for working closely with us with the ear tags.
Thank you very much, Sarah, for that presentation. Really insightful. Again, remember you can send questions through the Q&A chat. I'll probably kick off with one. Do you think we could be weaning earlier, given the monitoring of the rumination than what we currently do, or that the way that the tag measures rumination, tapping to the question for Lara before, might need to be adjusted for calves?
Yeah, so the interesting thing is just looking at weight gain post-weaning, not necessarily from our study or with the ear tag. We can see that our calves, actually, some calves can be weaned early because they're ready. Their rumen is ready earlier. And then we see that some calves need a little bit more time. Now, the problem with that is a lot of, farmers are weaning in groups, which is practical, from a management perspective. So the idea is that we'll be able to identify the calves that are ready earlier, and then the calves that need more time based on rumination, and maybe looking for a threshold. So that's future research, looking for a threshold that we can kind of determine when is a calf ready to be weaned.
Perfect. There's a question that came through. Hi, Sarah, very interesting work. How does early rumination impact long-term productivity? Any comment on that? I know you said it could be looked at, but any reference of the literature or thoughts?
That's a great question. I think there's quite a gap in looking at the impact specifically of rumination and development early on, moving forward to the production stage. So I think we won't be necessarily looking into that specifically, but I think that there's definitely a need for that for our industry.
Perfect. And the last question is, do you have any comments around the availability of hay and forage for these cows? And can the rumination be impacted by them consuming some of this and not only concentrate?
That's a great question. There's so much research out there that is looking at different feed regimens and the impact that it has on rumination and development. And I know that particularly the Australian industry is more pasture-based. So there's that kind of potential to really look into using this ear tag and the development of the calves, like incorporating different feed regimens into when to wean around using this technology.
Perfect. Sarah, thank you very much again for your time today, the video and the abstract, and we'll see you again in a couple of minutes.
Thank
So
you.
I want to call now our last emerging scientist, Ridong Xiang. Ridong obtained his PhD in animal genetics at the University of Adelaide in South Australia, and now he's working as a research fellow at the University of Melbourne, and also on a joint fellowship with AgVic. And he works on how to use multi-trait and multi-omics data to improve genomic selection of complex traits. So Ridong, no pressure, you're the last emerging scientist, so the stage is yours.
All right, can you hear me, Nicole?
Yeah, we can see you and we can hear
Cool.
you and we can see the slides.
Thanks very much, Nicole, for the nice words. Very happy to be here to share a talk on the customized DNA chip for accurate genetic testing of cows. So it's tailored to the Aussie cows, and it's just better. Now, the genetic testing forms an important part of Australian dairy farming, and essentially it predicts the future cow performances only using their DNA. Now, taking this as a simple example, that if we take some hair samples of the cows at site to the lab of genetics, and then we do some testing using this little plastic called DNA chip, where there are many, many markers on it, so we can get the counts of the alleles of a cow across its genome. And we use that data in combination with the phenotype data in database to predict the breeding values or performances of any cows. Like, for example, here we provide a prediction of the performances of cow milk yield per year, and this one, cow one, appears to be best, so we're going to recommend this to the farmers for breeding. Now, this is a prediction, so it's not perfect. So we look for ways to improve the prediction accuracy, and this little DNA chip sits in the center of this business. We believe this is the place we can improve the accuracy. Now, I want to use a little analogy to illustrate this process. Imagine we're actually scoping cow performances via lenses of a DNA. So here is a farmer, she looks at this non-prescribed lenses, wanted to know the performances of the cow. Now, because this is equipped with the standard DNA chip, which is not specific to any Australian cows or any phenotypes. So you could imagine that an image this farmer gets is rather blurred. So in our language, we'd say this is a low prediction accuracy of cow performances. Now, imagine there's another farmer who is using a prescribed lenses of the DNA to look at cow performances. And in our language, he or she is using this customized chip tailored to the Australian cow performances, traits, and populations. And in that scenario, we can imagine this farmer gets a clearer image of the performances of the cows. And in our language, that means high prediction accuracy of performances of cows. And that is really what we have been researching here to help the farmers get a better image of the cows' future performances. So my research is about tailoring the chip to the Aussie cows. We use advanced statistical methods in combination with functional genomic data to mine the genome-wide data, finding important markers for Australian cows. So once we find these markers, we're going to put them on this little plastic. We call this process customizing DNA chip. And this is actually look of the chip. And we're basically updating these little dots on each one of the chips. And that allows us to have a prescribed DNA lenses, if you will. They help us to look through them and get a better image of the cows. Now, this work is actually been implemented. So we have developed a new chip and used that to genotype about 15,000 Australian cows. And the latest results show that there's about 10% increase of the prediction accuracy for every cow we tested. Now, what does it mean for a farmer? Well, if we can predict the performances of cows better, that means in a database, we can rank the animals more accurately and more reliably. And that means we can provide this information to the farmers and they will get more accurate information to make better breeding decisions in terms of which cows to choose for breeding. And if they keep doing that, choosing the right cows, they're going to gain a lot of better genetics in a herd. So in the long term, they will improve their genetic selection of their herd. And that's good news for the farmers. There's more to that. Actually, we have implemented this genetic evaluation using the new chip for the Aussie Reds nationwide, and that implementation for Jerseys and Holsteins is underway. And also, our primary results show that this new chip not only works for Australian cows, but they work for the cows from the USA and New Zealand. So we are really here delivering a global impact of improving the genetics of dairy. And I'd like to wrap up with our the collaborators and funders who supported this work. And here is our publication on Nature Communications, and also a few media coverages of our work and more layman terms about this research. And that'll be all. Thank you very much, and happy to take questions.
Thank you, Ridong. I personally need to admit that probably this is the first talk related to genetics and genomics that I understand from start to end fully. So this to me speaks very highly of your capacity to transmit some of these findings and understandings. So thank you for that. I'm not voting today, unfortunately, but yeah, very well done there. So again, this is our last speaker. So this is the last opportunity to make any questions that you have. To kick off, probably, I've got a question around Are these chips that you found very different in either location or type to the normals that we were using before?
Yes, very different actually. So we have actually compared the overlap of the new chip and the old chip. So the overlap is about 10 to 20%. So we're talking about 70 to 80% of the markers used are different from the previous one.
Perfect. There's another comment saying they agree with me. Great presentation. There's another question saying, is the phenotyping only related to milk production, or did you look at other traits?
That is a good question. Well, so we did look 40 traits. So that's everything we have in the database. And so in theory, it covers traits like fertility or type traits. But because we have the largest number of records for production traits, whereas records for fertility or other type traits, they are kind of in a small number. So then while we looked for 40 traits, but in the end, we think that there is a bit of, higher weights towards the milk production traits, lower weights towards the other non-milk production traits. And this is something we are trying to improve in the next few years. Perfect.
Excellent. And probably the last question is from an industry perspective and an application of these findings. From a pharma point of view, kind of nothing would change. They would still need a hair or a tissue sample. It's more a methodology of kind of behind the scenes and lab that improves this, does it?
Yes, exactly. So we're just improving this process in a backstage. So it's just when you do the same thing, you'll just get hopefully better results. And there's also no cost increase because it's just the same number of markers and same chip. It's just we're changing the content of the chip using the new one. Yeah, that's right.
Perfect. Ridong, thank you very much for that, for your video and your abstract, as I told everyone. And we'll now wait till the end. So I would like to call to the virtual stage, if that makes sense, all the emerging scientists that presented today to turn on your camera. And I'll ask Lynn in the background that we will kind of kick off the voting. You need to select the emerging scientist that you think delivered the best talk or that you got more out of it. The emerging scientist and I cannot vote, so it's all up to you of choosing who you think was clearer, bigger impact, or something around that. In the meantime, I want to ask the 7 emerging scientists that are on the screen. As I told you at the beginning, Dairy Up is offering 9 PhD opportunities for anybody interested in doing research in a range of topics from pastures to cows to milk and the like. Who would like to give me kind of a thought of why should somebody consider doing either a master's or a PhD by research? And I know, having been in that path, it's got its ups and downs. We'll try to focus on the ups in this case, but does anybody want to give a thought of why should somebody consider doing some research and why it's kind of a good opportunity? Don't all jump at once.
I'll go ahead
Clara.
and start. Yep. I think that the best thing about Dairy Up is that the research is actually going to impact the dairy industry directly. So it's not only for yourself that you're making an impact, but it's for the industry. So I think that if you're going to do a PhD, I think you need to be passionate about the dairy industry.
Perfect. Anybody else kind of reflecting on their experience around doing research and working with industry to address an issue?
Yeah, just for me, I grew up on a farm, so I'm sort of really, I really love sort of the farming side of things and gaining a really strong understanding of the foundations of what you can learn as a scientist and sort of how these impact what farmers do, especially on the genetics front. Like we can implement change relatively easily through the breeding values and data genes. So being able to watch something that you do progress through the chain is quite amazing and very rewarding.
Perfect. Anybody else? A last minute reflection while we are voting? Any comments, any thoughts, any reflection of participating in this program for future emerging scientists?
Maybe I'll just comment a few. As from a data science point of view, there are actually a few other emerging scientists mentioned they're using all sorts of big data to address the issues in the dairy or in agriculture. And this is where you can really practice and learn about data science as well, which is an emerging field in industry and in science. So I think you can also learn a lot from that point of view. And there's a lot of data in the dairy industry, not just genetics, and also phenotypes and environment and everything. So it's a great place to learn and to improve.
Well, thank you very much, everyone, for sharing that. And we'll move towards the end of this session to announce the winners. So remember there was a scientific committee, and I want to thank Sabrina Lomax, who was also kind of coordinating this program with me. There was a scientific committee that evaluated the abstracts. Then there was a broader group that looked at the videos, and the audience here today voted for the best presenter. So I'll share the winners of each of those 3 categories. And I want to again acknowledge the sponsor of this session, Local Land Services. So the first prize for the best abstract that was submitted goes to Lara Hohen, that talked about her research on monitoring calf health using an accelerometer-based ear tag. So Lara, you're the lucky winner of $1,000 prize for that best abstract. So well done. The second prize goes to the best promotional video. So that was the 1-minute promo video that we used to advertise the DRF Symposium with the audience. And the best video goes to Juan Gargiulo, that developed a video on a decision support tool for robotic dairy farmers. So Juan, you're the lucky winner of a $500 voucher to spend with your supervisor and manager, which is really good. The third winner is the one that was selected today by all the audience, and I want to thank all the people that voted. First, I want to say we won't disclose the individual results, but a lot of people were very close. Everybody, for the sake of the emerging scientists, everybody got votes. Everybody got multiple votes, but obviously we need to determine a winner for this presentation today. And the selected winner of the presentation today is Beth Scott on does selecting for A2 milk increase inbreeding? And Beth is the lucky winner of another $500 voucher to spend however she wants, or to give to Juan that needs to give it to me. So with that, I want to thank you all again, the 7 emerging scientists, your supervisors, your research teams for all the great work, and the committee again for pulling this together. And I'll hand over back to Yanni Garcia to close the symposium today.
Thanks very much, Nico. And wow, what a session to finish off. That was incredible. I will just be very brief. I just want to thank again our sponsors. We cannot do this symposium without them. Local Land Services, our farmers, Dairy New South Wales, Dairy App, Dairy News, Bigger Cheese, and the Sydney Institute of Agriculture. I really want to acknowledge the organizing committee. There are too many to mention there, but I do want to highlight the terrific work by Lynn Garner from the Sydney Institute of Agriculture, and also the 2 additional help from Cara Jeffrey and Josie Clark, PhD students in agriculture at this university as well. So that their work behind the scenes was absolutely fantastic. Thank you very much, Lynn. I know they're not going to be showing your screen on, but you're most welcome to do it if you want. I'd like to also acknowledge all the speakers. I mean, 15 speakers overall within the 4 sessions. And as I said at the beginning, the 3 reasons for doing this is to disseminate our own research. You had an excellent example with Dairy App session, was terrific, and there's a lot more to come. The 2 sessions on risk management, farm management, and also people management, that was absolutely fantastic and with a lot of insight. And wow, what a session, the icing on the cake, in my view, the emerging scientist was absolutely terrific. And I don't know, I hope you all agree that we are in really good shape for the future of dairy research in Australia and overall. So thanks also to the chairs, to the 4 chairs, Niall Blair, Sabrina Lomax, Greg Duncan, and you, Nico, now. That was really, really appreciated. And a big thank you for all of you for attending this symposium. We hope to next one we can really welcome you face to face, and we look forward to that. And with that, I will finish this terrific 2 days. Thank you everyone for your participation and for supporting the Dairy Research Foundation. Thank you.